Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Claire Rougeulle from the Epigenetics and Cell Fate Center at Université Paris City on this show. Please let me briefly introduce you to our audience. You received your PhD in 1996 from Université Pierre et Marie Curie in Paris. You were then recruited to the CNRS in 1999 after your postdoc at Harvard Medical School in Boston. And you joined the Epigenetics and Cell Fate Center as a founding member in 2009. And you were the deputy director at the center since 2019. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Well, I think initially I was more interested in general in investigation, trying to make connection and link between things. And then when I was in high school, I saw a movie called The Hunter with David Bowie. And this is when I decided that I wanted to be to do science and probably to become a researcher. Actually, maybe more than before knowing that I wanted to do biology. And it was kind of a revelation. It's not really thought of, but something I really wanted to do. So I hesitated a bit, really a bit, between doing medical studies and biology. And then I went on for biology. And I really knew from the start that, as I said, I wanted to do research. So I didn't know in which field, but I knew that I would become a researcher.
So it was more the curiosity that drove you than the topic itself?
Yeah, exactly. Yeah.
Okay. So let's come to the science that you're now doing that centers around the epigenetic control of gene expression in relation to cell identity and fate, the contribution of long non-coding RNA genes and transposable elements to these processes, and the plasticity of epigenetic regulations in evolution. And all of this in the context of X-inactivation. I want to start with a paper that was published in the year 2009. So that probably was already from when you were PI. There you described that developmentally regulated transcription of six, I can't pronounce this, is required to block the spreading of the H3K27 trimethylation domain to the adjacent H3K4 dimethylation-rich XIST region. Can you talk about how you got into that and what you found there?
Okay, so actually, I become interested. So I started my study on X-chromosome inactivation during my PhD, actually, which was a long time ago. And at that time, we knew very little regarding the molecular mechanisms and even the actors of the process. So at that time, I was really interested in deciphering, physically mapping the region that controlled X-chromosome inactivation. So it was way before anything related to chromatin modification was considered. And then after my postdoc, I came back and started to look at really chromatin-related events in X-chromosome inactivation and in the role that non-coding RNA the role that non-coding RNA could play in this process. And that's when we became interested in the non-coding RNA called TSIX, which is the antisense to the long non-coding RNA XIST. So XIST is the trigger of X-chromosome inactivation, and TSIX is an antisense that does a number of things, but in particular, we control the deposition of chromatin mark within the XIST locus, and which we believe control to some extent the choice of which X chromosome will upregulate XIST. And then there is this other long non-coding RNA called FTX. Is it the one that you are referring to in this 2009 paper?
Yes, also, yeah.
So this is this was really the beginning of the story when we started to realize that X inactivation was not only about XIST and its antisense TSIX, but that there was also other long non-coding RNA coming from the same locus called the X inactivation center. And at that time, we had discovered this new or recently discovered FTX, which was sitting, or the gene was sitting in a region that we had previously identified as being enriched in chromatin repressive marks. So those marks that are hallmark of the inactive X chromosome, but these marks were present in this region before X chromosome inactivation was initiated. So we thought that maybe they could play a role in nucleating kind of an heterochromatic domain. And so we were interested in understanding to what extent this TSIX, this FTX non-coding gene could be involved in this nucleation.
How does this relate to XACT? Because you also reported the discovery of this long non-coding RNA. How are they related to each other?
So they are not really related, actually, beside the fact that they are coming from the X chromosome. So initially, most of the studies done by a lot of people working on X inactivation, as it was the case for us, were done using mouse as a model. And that's where we started to understand, the function of XIST and then the role of other long non-coding RNA genes in regulating XIST expression. And then I think it was in 2008, or no, even a bit before actually, I really was curious about what would happen in human, because we knew very little about the way X inactivation would be regulated in human. So that's when we decided to include humans in our studies. And for that, we used human embryonic stem cells that were available at that time. And so we took kind of a blind approach, looking at by just RNA sequencing at what was happening in human embryonic stem cells, which corresponds to early developmental stage where X inactivation is being initiated, let's say, and compare that to more differentiated human cells. And so we did analyze the data in a manner that allowed us to identify some transcripts that were not previously identified. And we came across a number of transcripts. So we were interested in transcripts coming from the X chromosome. And we were interested in transcripts that were expressed in human embryonic stem cells, but not in differentiated cells. And we identified few of them. And because what we like to do then is to look at their expression at the single cell level in the nucleus by RNA FISH, so fluorescence approach. Then we looked at all these new transcripts that we had identified. And one of those was really peculiar in the sense that the RNA was found to form a cloud in the nucleus. And I really recall this moment where actually it was Céline Vallot in the lab who did this experiment. And she was at the microscope and she called me and she said, "Oh, Claire, you better come. I think we have something exciting there." And this was a nice moment in research where you become really, really excited. So that was way later than these FTX stories, but it was really exciting.
So what was it that she saw there exactly?
So what she saw is that in those human embryonic stem cells, where the one X chromosome is already inactivated. Of course, if you talk about female cells, then the inactive X chromosome is decorated by the XIST long non-coding RNA, so XIST being the trigger of X chromosome inactivation. And what she saw was that on the other X chromosome, which was thus the active X chromosome, the XACT long non-coding RNA was also forming a cloud, so accumulating around the chromosome. So we had kind of a mirror image with one X chromosome, the inactive one, being coated by XIST, long non-coding RNA, and then the other chromosome being coated by this novel long non-coding RNA, which we call XACT for X-active coating transcript.
Yeah, I also remember back in the day when I was doing immunofluorescent and fish that when I did the H3K27 trimethylation staining, you really could see those bar bodies or those inactive X, and they were really nicely stained and also located somewhere else than the other ones, which is really nice to see. What you also can do, or the epigenetics field has more players than just long non-coding RNA and histone modifications, but then there are also transcription factors. And what you then looked at was the transcription factor yin-yang 1, so YY1, in the transcriptional activation of XIST in both human and mouse. So was this then the next logical step, or how did you get into that, and what did you find there?
So this was a story that started a long time ago, because there was a moment in the field where it was thought that XIST was mainly accumulating through post-transcriptional mechanisms, so stabilization of the transcript. And so back then we were interested in understanding whether XIST could be also regulated at the transcriptional level. So I would say it's not really epigenetics, it's more like transcriptional regulation. It happens to be the transcriptional regulation of an epigenetic actor, but in itself, the process is rather transcriptional regulation. And so we wanted to know which were the factors that could contribute to this transcriptional upregulation. And looking at the sequence, we had identified within the first exon of XIST, so in the 5' region of XIST, some binding site for, let's say, key factors such as CTCF, which is also an architectural protein or factors, but can also play a role in transcriptional regulation, and YY1. And so this was done essentially in the mouse to begin with. And so we did some, let's say, functional analysis looking at what was the effect if we would either knock down YY1 or delete the binding site, their binding site within XIST. And this allowed us to show that indeed YY1 was important in promoting the transcriptional activation of XIST in the mouse. And then by doing a sequence comparison, we could see that those YY1 binding sites within the first exon of XIST were found in all placental mammals that we looked at, where XIST is conserved. And so we assume that this mechanism of transcriptional upregulation of XIST by YY1 would be conserved, which we validated functionally, experimentally to some extent in human.
So it's present both in human and mouse. That was the bottom line.
And it seems to be a conserved mechanism for XIST upregulation.
So maybe we can take a little detour here, because you just said that epigenetic, or you used the terms epigenetics and transcription regulation as like two different things. So where do you draw the line, and what is the difference for you of those two terms?
So for me, epigenetics refers to mechanisms that indeed maybe control the expression of genes, but with a memory component. So something that would be able to memorize an initial. So there would be an initial stimulus that would dictate a transcriptional response, but then this response would become autonomous independently of the original stimulus. And actually, when you think about this definition, I'm not too sure that there are so many epigenetic mechanisms. But this is a subject in itself that would take hours to discuss.
So transcription regulation would then be transcription factors, and epigenetics would be histone modifications and DNA methylation, or?
I think it's even more complex than that, because it's not because to me, it's not because chromatin is involved that it's epigenetics. It would become epigenetics if the chromatin can be self-memorized during replication, but I don't think it has been that well demonstrated. So I mean, even in the case of X-chromosome inactivation, where you do have, of course, chromatin modification of the inactive X with some repressive chromatin marks, such as H3K27 trimethylation, which is supposed to be epigenetically transmitted. If you get rid of XIST, then these chromatin marks disappear, so they cannot be maintained by themselves. They need to be recruited indirectly by XIST. So, this is a wild field of discussion. What is epigenetics?
Definitely, yeah. So coming back to topic, what you then did was looking at the erosion of X-chromosome inactivation in human pluripotent stem cells and how this might be influenced by XACT. So can you maybe explain what the erosion of X-chromosome inactivation is and what you found there?
So I think, X-chromosome inactivation is something, I think it's artifactual. So I don't think it has an obvious biological relevance. This is being said with cautious. Being cautious, yeah. So the erosion is the fact that if you keep human embryonic stem cells or human pluripotent stem cells in general in culture, so these cells are usually in a prime state of pluripotency, meaning that they correspond to post-implantation epiblast, so they do have already an inactive X chromosome. But what happens is during prolonged culture of these cells, there are several hallmarks of the inactive X that are lost spontaneously in culture. So XIST start to be repressed. Some repressive chromatin marks that are deposited by XIST are lost from the inactive X chromosome. And some of the genes on the previously inactive X chromosome become reactivated. So this is, as I said, something that occurs spontaneously in culture that may impact or at least the use of those cells as a model system for early development.
Does it happen because the external stimuli are missing, or why does it happen?
I don't think it's that clear actually. Why is it happen? Either the like a selection of the cells, that the cell that would tend to lose those marks of X chromosome inactivation would have like a higher proliferation rate, and they would outcome the cell that keep the one inactive X. So this is something that we wanted to explore at some time, and then we didn't go that far. But so even though it's artificial, we thought we could still maybe learn things about from erosion, because we thought this is something that happens with pluripotent cells, but not with other cells that are more differentiated. So I think we thought it would maybe reflect a peculiar state of the inactive X chromosome at this stage that would really relate it to the fact that we are working with embryonic cells and not more advanced cells. So that's when we wanted to understand what was the origin of erosion, coming back to your question. And so we looked at different markers, and especially XACT that we had just discovered at that time. And so what we found is that the expression of XACT, so initially XACT is expressed only from the active X chromosome, which codes. And then then ultimately, when the cells undergo erosion, now XIST is no longer expressed, and now XACT is expressed from the two X chromosomes. And so we try to capture the sequence of events between those two extreme states. And what we found was that XACT seemed to be one of the earliest events of erosion, because we could see re-expression of XACT from the inactive X chromosome before XIST was disappearing. So we suggested at that time that maybe the re-expression of XACT could play a role in the erosion process in itself, but it's only an hypothesis, and it has not been formally demonstrated.
So you also didn't check some other factors like the chromatin environment or things like that?
Not in a kinetics manner. So we could not we know that when XIST gets repressed, the repressive histone mark are lost. I mean, some of them, the ones that are triggered by XIST. But what comes first, this would be difficult because this is an asynchronous process. So we would need to look at that at the single cell level. And we haven't done that carefully, but there is, I would say that the repressive mark are lost following the disappearance of XIST, but this needs to be firmly looked at. Yeah.
In the beginning, you said that you were looking at single cells already because microscopy is, per definition, a single cell method. But what you also did was then identifying human-specific mechanisms regulating X-chromosome activity in early embryonic development by single cell imaging and single cell RNA-seq. I mean, the development of those single cell methods by sequencing would probably be very helpful for you if you are coming from the single cell microscopy field, right?
Actually, yes and no. Because yes and no, because to look at X chromosome activity at the single cell level by sequencing approach is not trivial, because the way to look at X chromosome activity requires to be able to distinguish between the two X chromosomes. So this relies on the presence of SNP, single nucleotide polymorphisms. And so you need to be able to distinguish the presence of SNPs within a single cell. And this is, as I said, not trivial, because most of the single cell sequencing approach are based on 10x sequencing, which is the sequencing of the 3' end of genes. And you might expect those 3' end of transcript actually to be enriched in SNP because these are untranslated region, but actually there are not that many. So you don't have a lot of informative position by looking at 10x genomic data. So meaning that ultimately there are not that many transcripts that you can assess in terms of allelism. So you would need to do full-length sequencing. And even that, because when you do single cell sequencing, you don't have that many reads per cell. You lose a lot of informative positions. So it's not easy to have a comprehensive landscape of the allelic expression of X chromosomes. So this is really a challenge in the field, actually, to be able to assess allelically the expression status of a number of genes on the X chromosome.
Are there ideas to overcome this? Other than, I mean, you just said that there are SNPs, but I mean, that's also
so
like
the
a technical...
ideas, yes, so there are some ideas, which is, so I'm not sure that's going to be doable at the single cell level, but if we move to the analysis of chromatin landscape by looking, for instance, at the distribution of H3K27 methylation, which is marking the elongating polymerase, so the whole gene bodies there, because it's marking introns as well, we will probably find more polymorphism than just by looking at exons. So at least by bulk RNA sequencing, we will have more information. By a single cell, it's going to be more difficult. And then other approach would be probably to develop imaging approach that would allow to probe expression of multiple genes, just not few of them, but a large series of them in a more systematic manner. I think that would be the combination of those approaches that may bring new information. Yeah.
What you then did was you identified a critical enhancer for the regulation of the XACT locus that evolved from an ancestral group of mammalian endogenous retroviruses prior to the emergence of XACT. So this again plays into the role of evolution. Can you talk about this study and what you found?
Yeah, so the XACT locus was quite puzzling in the sense that, so it's a very long non-coding RNA. At the end, it's more like 250 kilobase long transcript. So it's quite a long non-coding RNA. And it is aminoid specific transcript. And so we were interested in the way this sequence emerged. And so we found out that the promoter of XACT was emerged through the insertion of a transposable element. So an LTR5. And this is only in aminoids. And then we quite early on, when we discovered XACT, realized that the expression of XACT was mainly restricted to pluripotent contexts. So we wanted to understand what was driving actually this restricted expression profile. And so we tried to look at regulatory sequence in the vicinity of XACT that could drive this pluripotent specific expression profile. And so we found a region where which was bound by pluripotency factors, so OCT4 and SOX2 and NANOG, if I recall well. And so we deleted this region and confirmed that indeed it was promoting the expression of XACT. So this was probably what was linking XACT expression to the pluripotent context. And so we looked more into this sequence, and it turned out that this sequence that binds those pluripotent transcription factors is in fact retrotransposon itself, but of a different family. And it's a family that has invaded primate genomes before the family that form the promoter of XACT. And so it was conceptually interesting in the sense that it was as if this sequence was already there, and it was already bound by transcription factor in species such as macaque, where XACT was not there. And then later on, during hominoid speciation, there would be invasion by other families, including so these LTR5, and that would somehow hijack this regulatory sequence that maybe had other function in these species, and hijack this sequence to now regulate this newly emerging long non-coding RNA. So that was conceptually quite interesting, we believed.
Did you also look at the local chromatin environment? So how, if it formed a loop or whatever there was?
So there seem to be, yes, loops between this region and XACT, and there's also another locus that evolved concomitantly to XACT, which we call T113. This is based on the actually coordinate of the locus on the X chromosome, and there was indeed some looping occurring between this sequence and XACT. But to give the full story about XACT, so obviously the big question is, what is the function of XACT? So this is something that we were keen to address from the beginning, but this was a challenging question to address initially because we thought it could be playing a role before inactivation was initiated. And so by somehow preventing XIST from silencing the chromosome. So I don't know if this is a question that you had planned to ask later on, but
yeah, you can go into that now.
Okay, so
please go ahead.
So when we looked, when we became interested in XACT, of course, this had been identified in cells in culture. So we wanted to know whether this was an artifact or whether this was somehow physiologically relevant. And the only way to address this was to look at human embryos. And so we could have access to human embryos. We collaborated with Catherine Patra at that time. And so we could look at XACT expression in human pre-implantation embryos. And what we found was that in those embryos, it is known that this is something specific to primates, or at least which is different from rodents. In pre-implantation embryos, the two X chromosomes in female embryos are active. So there is no X inactivation undergoing there, in contrast to the mouse. So what is quite striking, and that was found by the lab of Edith Hurd, is that they are both actives, but they are both coated by XIST. So this is puzzling because XIST is known to induce silencing of the chromosomes that it coats. So how come you could have two X chromosomes decorated by XIST yet being active? So this was really a question that we wanted to address. And so by looking at XACT, we found that XACT was also decorating those two X chromosomes, and that the XACT RNA and the XIST RNA were not at all overlapping. So they were repulsing each other. So we came with the idea that maybe XACT would be somehow pushing XIST out of the chromosome and preventing maybe XIST from actually silencing the chromosome. But to be able to formally address this question, we needed to have an in vitro system which would recapitulate the status of the embryo. And this is not what is happening in primed human pluripotent stem cells. So we couldn't do that initially, but more recently, there were protocols that were developed that reset human embryonic stem cell into a naive state of pluripotency. So a state where the two X chromosomes are active and decorated by XIST. This is not absolutely perfect, but let's say it's like this. So decorated by XIST and XACT. So with these cells, now we could address the function of XACT. So the idea is you delete XACT, and if it is preventing this from silencing the chromosome, now you would be able to see X inactivation in naive human embryonic stem cells. So we did this experiment and nothing happened. So the X didn't get silenced in the absence of XACT. So this is showing that you might have interesting hypotheses and they might prove wrong. So for the moment, we have no idea whether XACT has any function. We are still exploring other possibilities. But actually, the first hypothesis that we had was not verified experimentally.
Okay. Yeah, this leads to my next question that I wanted to ask, because we now talked about what you did up until now, but what is it that you're focusing now and for maybe, let's say, the next 5 years? So the question is, imagine you're due to submitting a grant proposal tomorrow. What would you have written into that?
This is highly confidential. Why should I tell you that?
Yeah.
No, I'm kidding. So there's various aspects that we would like to study. So one is understanding how X inactivation is established in human, because it's radically different from everything we know in the mouse. So in the mouse, we know that, we don't know everything yet, but we know that X inactivation is established by the upregulation of XIST that would recruit a series of factors that promote transcriptional silencing, chromatin reorganization. So, there are still many questions left, but we start to have a rough idea about how the process is established in the mouse. Whereas in the human, what happens is that we start from a situation where XIST is sitting on the two X chromosomes. It is not silencing the chromosome, but it is actually attenuating the expression of the two X chromosomes. So this is something that we have shown and that the lab of Catherine Plath has also shown. And then at some point after implantation, there is a transition from a state where XIST is expressed from the two X chromosome and just dampening their expression, from a state where XIST is expressed only from one X chromosome and now fully silencing this X chromosome. So how do we transition from between these two states? What mediates this shift in XIST activity? This is totally unknown. Whether this is established in a cell-dependent manner, so there is evidence that probably this occurs first in extraembryonic cells, but whether this occurs simultaneously for all the cells, simultaneously for all the genes on the X, and to what extent the inactivation of one X chromosome is linked to the human development, this is something that is not understood and that we would like to explore.
Mm. So for the last 36 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
So we talked a lot about, the science, but what is important, I think more important are the people who do the science. So it's clearly not me. So the people are all the people that are working with me. And this is, I think, the most beautiful part of the journey, to exchange with people, to train people, to exchange ideas and so build together the project and help also to train the people, to train the next generation of researchers. I think that's what is even more stimulating than the science, because it's a daily pleasure, while the science is sometimes a bit frustrating. So I think, but this is the beauty of being a researcher, is that you have many different aspects to your work that is making it highly stimulating.
But your job is to provide the framework, right? And the money and all the right tools for them to shine.
Of course, I hope and I tend to believe that I'm still useful in the lab. So providing the framework and also trying to stimulate the people and giving some direction, but it's also extremely important to have the feedback from more experienced researcher. I'm lucky to have as collaborators, and so that's really the mixture of all our maybe talents or motivation that makes those things work. So it's important. It's not a one-person story. Definitely not.
So back in the day when I was doing my PhD, the boss of the institute, he always came to the lab like right before Christmas to do one experiment. And this was his famous Christmas experiment. Do you also have some kind of habit of this, going back to the lab?
No, I did that for a while. And honestly, it was very frustrating for everyone, I think. I mean, first for me, because I could start the experiment, but I could never finish it or analyze it properly. So it was a waste of time and money, and probably for the entire lab, because I was always complaining about not knowing where things were and so on, and asking help from people. So I think, I have fully stopped doing experiment, and I'm okay with it.
So thank you, Claire, for your time and for being on the show.
Thank you very much for this invitation.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.